The interbreeding of closely related sympatric species of animals is prevented by a whole series of ecological, behavioral and cytogenetical factors, usually several for each species pair. One factor is often dominant, such as acoustic stimuli in certain frogs, grasshoppers, cicadas and mosquitoes, chemical contact stimuli in some insects, sex stuffs in Lepidoptera and certain marine organisms Bonellia, etc., and visual stimuli in certain birds, fishes and insects. The sterility barrier may be strong or weak. Boot is rarely tested except when the other isolating mechanisms break down. Thus, the isolating mechanisms are arranged like a series of hurdles. If one breaks down, another must be overcome. For example, if the habitat barrier is broken, individuals of two species may still be separated from each other by behavior patterns. If these also fail, the mates may be unable to produce viable hybrids, or if hybrids are produced they may be sterile. See Meyer, 1970. Isolation 123. The genetics of isoiating mechanisms. From the multiplicity of isolating mechanisms that protect every species, one can conclude that a considerable number of genes are involved. Almost any gene that changes the adaptation of a population may have an incidental effect on the interaction between male and female. Conversely, a mating advantage of a male will become established in a population provided it does not seriously lower the fitness of his offspring. Detailed analysis of the genetics of isolating mechanisms other than sterility exist only for the genus Drosophila. Many mutations in D. melanogaster, such as yellow, white, and bar influence the mating success of the mutant individuals. Further studies have established now that behavioral isolation has a highly complex genetic basis. In addition to genes controlling the specificity and variation of the signals exchanged between males and females, there are others that control mating drive and levels and rhythms of activity. ROIEOFISOIATING mechanisms. One of the evident functions of isolating mechanisms is to increase the efficiency of mating. Where other closely related species do not occur, courtship signals can afford to be general, nonspecific and variable. Where other related species coexist, however, nonspecificity of signals may lead to wasteful courtship and delays, even where no heterospecific hybridization occurs. Under these circumstances there will be a selective premium on precision and distinctiveness of signals. This is exemplified by Peru's bird of Tenerife Island and Regulus songbird of Azores. Moreover, each species is a delicately integrated genetic system that has been selected through many generations to fit into a definite niche in its environment. Hybridization usually leads to breakdown of this system and results in the production of disharmonious types. It is the function of isolating mechanisms to prevent such a breakdown and to protect the integrity of the genetic system of species. Any attribute of a species that would favor the production of inferior hybrids is selected against, since it results in wastage of gametes. Such selection maintains the efficiency of the isolating mechanisms and indeed helps to perfect them. Isolating mechanisms are among the most important biological properties of species. Origin officiation. The following two theories have been proposed about the origin of isolation. Wallace has proposed the first theory. According to him, isolation mechanisms are selected by natural selection to root out lowly fitted hybrids between species and allow only those genotypes that have developed better isolation mechanism. There are instances where hybrids survive better under vigorous environmental stresses than the pure races. The serious objection to this theory principally comes from Darwin. If the back-crossing and introgression by a certain hybrid population would lead to the steady-state deterioration of the pre-existing mechanism. Thus, Wallace's argument of improvement of isolation is failed. Poulton and Muller put forward the second idea regarding origin of isolating mechanism. According to their idea, isolation mechanisms originate as a byproduct of genetic divergence of isolated populations. Genetical studies reveal that hybrids between two new species must be effectively sterile or inviable. Natural selection then picks out such instances, improves accessory isolating mechanisms, such as failure of sterile species survival. Such a selection ultimately renders hybrid production lesser and lesser in nature. RUAYRYO 018 DMN OK. Speciation. The entire course of evolution depends upon the origin of new populations, species, that have greater adaptive efficiency than their ancestors. The study of population divergence or speciation is, therefore, crucial to an understanding of evolution. Before the discussion of speciation process, a brief discussion of the term species is desirable. Species, race and deme. A species may be defined as an interbreeding population, which is reproductively isolated from other similar but morphologically distinguishable populations. In other words, a species may be envisioned as an isolated pool of genes flowing through space and time, constantly adapting to changes in its environment as well as to the new environments encountered by its extensions into other geographical regions. At various intervals of time and in different environments of this gene pool may become isolated. Such isolates may 1. Become extinct, 2. Reunite with the parental species, or 3. Differentiate during isolation to form new species which in turn pass through space and time. Certain general characteristics of animal species are the following. Each species is an isolated pool of genes possessing regional, racial, populational characteristics in gene complexes which are interconnected by gene flow. Each species fills an ecological niche not exactly utilized by another species. These species of cichlid fisher have descended from a single ancestral population within a million years. Speciation 125. Each species remains in process of continually adjusting to its environment. Each species possesses a constellation isolating mechanisms that directly or indirectly prevent exchange of genes with related species. b. Each species has the capacity to give rise to new species provided some form of geographical or spatial isolation gives its isolates an opportunity to develop a unique gene pool without being swamped by gene flow from the parental species. Lastly, a species is a population of individuals with similar structural and functional characteristics, which have a common ancestry and in nature breed only with each other. It is a collection of demes, the smallest population units, which are the groups of genetically similar individuals bearing an intimate temporal and spatial relation to one another. Each deme is isolated to some extent from adjacent demes, but there is each species remains in process of continually adjusting to its environment. Always the possibility of genetic exchange between them. Even distantly located demes may contribute genetic material to one another over a period of time by the gradual passage of certain genotypes from one deme to another. Thus, demes are open genetic systems that are affected by gene flow from adjacent populations, that is, they are only partially isolated populations. An intermediate population unit which exists in between species population and local population, deme, is subspecies, or geographical race or race. A subspecies is an aggregate of local populations of aspecies inhabiting a geographic subdivision of the range of the species and differing taxonomically from other populations of the species. The nature of species, subspecies and deme now arms us to solve the following questions. How from a single stock of population arise different species? Or, in other words, how does speciation occur? Nature of speciation. Speciation means the formation of species. Meyer, 1970. Ever since the speciation process became a separate topic in the 1930s. Meyer, 1931. Alternative. Ever since the speciation process became a separate topic in the 1930s. Meyer, 1931. Alternative methods of speciation have been proposed. The principal ones are quantum evolution proposed by Simpson, 1944, quantum speciation defined and discussed by Grant, 1963, 1971, and Carson, 1973, 1975, and called saltational speciation by Ayala, 1975, parapatric speciation discussed by Murray, 1972, and Bush, 1975, and sympatric speciation described by Stebbins, 1950, Clausen, 1951, Meyer, 1963, 1970, and Grant, 1971. The true meaning of the term, origin of species, used by Darwin was understood only rather recently. The pre-Darwinian evolutionists did not understand its exact significance. Even Darwin himself seems to have considered origin of species the same as evolution. He confused two essentially different problems, subsuming them under the single heading, origin of species. One of these is evolutionary change, in which Darwin was primarily interested and for which Darwin used the following terms: phyletic evolution, modification in time, or descent with modification. 126 evolution, evolutionary biology. Phyletic evolution means evolution in a line or lineage, species living in a certain region in the course of time undergoes change so that the descendants are sufficiently unlike their distant ancestors to be considered a different species. Mesohippus evolving into Myohippus may be taken as an example of such phyletic evolution in which species A has become species B. Typically, species A will have disappeared in the process, leaving species B in place. Similarly, as time goes on, species B may evolve O into and be replaced by species C and so on. This phyletic pattern of evolution along a time axis is abundantly evident in the sequences of fossil forms. According to Meyer, 1970, such evolutionary transformation of a species does not necessarily lead to multiplication of species or speciation. Because an isolated population on an island might change in the course of time from species A through B and C into species D without ever splitting into several species. Consequently, in the end there will be only one species on the island, just as at the beginning. The essential aspects of phyletic evolution is the continuous genetic and evolutionary change within the populations composing the species, without the development of reproductive isolation between subspecies or demes of a species and, consequently, without its breaking into several species. Phyletic evolution. Speciation in its restricted modern sense, however, means the splitting of a single species into several, that is multiplication of species. The opposite of splitting of a single species into several daughter species is the complete fusion of two species. Since the product of such a process would be an entirely new unit, truly a new biological species, it is legitimate to consider the fusion of two species into one as a form of speciation, which is the reverse of a multiplication of species. Since species are defined as reproductively isolated populations, the fusion of two species is, on the whole, a logical contradiction. Yet occasionally in nature a previously existing reproductive isolation breaks down and two previously distinct sympatric species merge. This happens most easily in species in which the isolating mechanisms are primarily ecological. Such fusion method of speciation has been reported in two species of birds, Tauges pipilo-erythrophthalmus and Piacae, both of which fuse because of breakdown of isolation and form a new species. In this chapter, a detailed discussion will be made only for speciation by multiplication of species as other methods of speciation, namely, transformation of species, phyletic speciation, and reduction in number of species, fusion of two species, are of little significance. Potentialmodes of speciation. According to Meyer, 1970, true speciation or multiplication of species may occur by the following agencies. Speciation 127. A. Instantaneous speciation through individuals 1. Genetically. A. By single mutation in asexual species. B. By macrogenesis. 2. Cytologically, in partially or wholly sexual species. A. By chromosomal mutations or aberrations, translocation, etc. B. By polyploidy. B. Gradual speciation through populations. 1. Geographical speciation. 2. Sympatric speciation. A. Instantaneous speciation. The process of instantaneous speciation may be defined as the production of a single individual, or offspring of a single mating, that is reproductively isolated from the species to which the parental stock belongs and that is reproductively and ecologically capable of establishing a new species population. It can be achieved by the following methods. 1. Instantaneous speciation through ordinary mutation. Mutation is a genetic phenomenon of such relatively high frequency that in a higher animal with more than 10,000 gene loci almost every individual will be the carrier of a new mutation. Such mutations merely increase the heterozygosity of a population, they do not lead to the production of new species. Any mutation drastically affecting reproductive behavior or ecology will be selected against, if it lowers the viability, or will displace the original allele if it is of higher viability. In neither case will there be any origin of discontinuities. It is evident that ordinary mutation cannot produce new species in sexually reproducing species. Certain phenomena among rotifers, cladocerans and nematodes suggest the occasional occurrence of asexual speciation by mutation. For example, in the rotifer order Beteloidea, there occurs no male individual in entire order and so it is assumed that the ancestral species of this order was parthenogenetic. This ancestral species evolved into over 200 species, about 20 genera and 4 families. Only due to asexual speciation by mutation. Vegetative reproduction by growth and splitting, thus, might offer a favorable condition for instantaneous speciation. This form of reproduction, however, occurs only in some of the lowest groups of animals, such as sponges, colinteраtes, hydroids, terbellarians and bryozoans, groups in which the taxonomy is as yet too uncertain for a study of speciation. Colinteраtes, hydroids. 2. Instantaneous speciation through macrogenesis. The sudden origin of new species, new higher taxa, or quite generally of new types by some sort of saltation, a change by leap across a discontinuity, has been termed macrogenesis. The macrogenesis theory of speciation has been supported by some geneticists such as Goldschmidt, 1940, 1948, and Schindwolf, 1936, 1950. According to this theory, the production of a new type by a complete genetic reconstruction, macrogenesis, or by a major, systemic mutation, or macromutation is the crucial 128 evolution evolutionary biology event in speciation. Such an event will produce a hopeful monster, as Goldschmidt, 1940 called it, which will become the ancestor of a new evolutionary lineage. The believers of macrogenesis theory also propose that all new types appear in the fossil record suddenly and abruptly. These types are not connected with the ancestral types by intermediates, they claim, and cannot be derived from them by gradual evolution. All these views of macrogenesis theory have been bitterly criticized by many modern evolutionists. The basic objection to macrogenesis theory is that it can never be proved by any kind of evidences, because it is obviously impossible to witness the occurrence of a major jump, particularly one that achieves simultaneously reproductive isolation and ecological compatibility. Further, Simpson, 1953, and Wrench, 1960, have criticized this theory by showing paleontological evidences which clearly suggest that one type can be derived from a previously existing one. Moreover, modern genetic studies have shown that the production of a monster or freak individual due to mutation does not provide the raw material for speciation, as such freak individuals have low viability, do not get any mate for sexual reproduction and subsequently fail to establish reproductive isolation from normal members of the parental population. Therefore, macrogenesis is a poor source of speciation. Meyer, 1963, 1970. 3. Instantaneous speciation through chromosomal aberrations. Closely related species often differ more conspicuously in their karyotype than in their morphology. Among aspects of the karyotype that differ are chromosome number, the number of metacentric or acrocentric chromosomes, the presence and kind of paracentric or paracentric inversions, or of supernumerary chromosomes, and just about every aspect of chromosomal aberrations. Visualizing this fact, most cytogeneticists believe that chromosomal mutations have a significant role in the causing of instantaneous speciation. This belief is based on two assumptions. 1. That the degree of difference displayed by two species requires a speciation process of such drastic dimensions that only chromosomal mutation can qualify, and 2. That reproductive isolation between two species cannot be achieved without chromosomal reorganization. Now, it is known that chromosomal rearrangements in most cases does not produce new species, however, only in few cases it produces speciation. Chromosomal rearrangement without speciation. Except deleterious chromosomal mutations, most kinds of chromosomal aberrations, mutations, lead to chromosomal polymorphism rather than to the development of isolating mechanisms. The paracentric inversions of Drosophila are a well-known example of such chromosomal polymorphism. Yet each species has its own specific polymorphism, and only very rarely do even the most closely related species share the same chromosomal polymorphism. This fact underlines the drastic nature of the chromosomal aberration during much of speciation. This phenomenon suggests that there is no necessary correlation between chromosomal mutations and speciation, since either can occur without the other. Speciation coinciding with a chromosomal mutation. If the chromosomal mutations during speciation would not have some selective evolutionary advantage, they would not have occurred so frequently in nature. Consequently, they have the following two advantages in chromosomal speciation. A. Chromosomal mutations have the potential to serve as or contribute to isolating mechanisms, and B. The locking up and protection of a particularly favorable gene complement through achromosomal mutation may create a new supergene, as Wallace, 1959, first of all recognized it. Both of these components of chromosomal speciation can subsequently be improved by natural selection, either during a period of segregation in a geographic isolate or during subsequent parapatric speciation, or by both processes. The term parapatric stands for the population or species which remain geographically in contact but not overlapping and rarely or never interbreeding. Meyer, 1970. Chromosomal mutations as potential new isolating mechanisms. Any change in the structure of the chromosomes is called chromosomal mutations or chromosomal aberrations, whether Idison inversion, translocation, duplication, or any other change in the linear sequence of the genes. Speciation 129. Or in the mechanics of the chromosome, for instance, spindle attachment. Chromosomal mutations, mostly inversions, are estimated to occur at a rate of 1 in 1,000. Most of these are sufficiently deleterious to be eliminated at once, that is, before the mutation's carrier can reach reproductive age. Other kinds of chromosomal mutations are capable of giving rise to a system of balanced polymorphism, and they will be retained in the population. In addition, there is a third class of chromosomal mutations which appears to reduce the fecundity, a term used for reproductive potential as measured by the quantity of gametes, particularly eggs, produced, of the heterozygotes to some extent. The heterozygotes containing some kind of chromosomal mutation that seems important in speciation, usually encounters the following difficulties during meiosis: gametogenesis, I. Meiotica synapsis, partial failure of chromosome pairing, or E. malorientation of multivalence at the first meiotic metaphase, or both. Both of these difficulties lead to the production of gametes carrying chromosomal deletion or duplication or broken or acentric chromosomes and, thus, lead to a significant reduction in the fecundity of male hybrids. Unlike gene or point mutations, chromosomal mutations usually have no effect on the visible phenotype of the individual, so they are difficult to detect. Further, unlike the heterozygotes containing point mutations, the heterozygotes of chromosomal mutations are not shielded by dominance, and such heterozygotes with their meiotic difficulties are the prime target of selection. John and Lewis, 1969. Finally, chromosomal mutation is not a change in the DNA program, but a change in the linear sequence of genes, duplication or a deletion. The most frequent chromosomal change is the fusion of two acrocentric chromosomes into a single metacentric chromosome. The result is the reduction in chromosome number. A trend towards such a reduction is widespread among animal taxa. For instance, the most primitive isopods have 28 haploid chromosomes, and this number is independently reduced through fusion to the lower number of 8 in several unconnected lines. It is now clear that most differences in chromosome numbers between closely related species of animals are the result of such fusions. If only fusions have occurred during evolution, soon all species would have only a single pair of chromosomes. Obviously, there should be some opposing mechanism which may increase the chromosome number. The seemingly simplest such process would be the exact reverse of fusion, that is fission or dissociation of a metacentric chromosome into two acrocentrics. Most cytologists doubted such fission to occur. However, increase in the chromosome number, not due to polyploidy, whatever the mechanism, is frequent in animal evolution and may well play an important role in speciation. See Meyer, 1970. B. Chromosomal mutation and the production of new supergenes. It becomes now clear that gene contents rather than mechanical qualities, as discussed in preceding heading, determine in many cases whether or not a new gene arrangement can establish itself in a population. Most structural rearrangements of chromosomes inhibit or prevent crossing over in heterozygotes. A new gene arrangement may lock up a codapted gene sequence and, by protecting it from crossing over, create a new supergene. Meyer, 1963. Wallace, 1959, pointed out that bearers of such protected chromosomes or chromosome segments, being members of peripheral populations, are better adapted for the marginal environment of the species border than are genotypes from center of the species range. To him, the most important aspect of the chromosomal reorganization is the protection from disruptive recombination that it affords certain new supergenes. Chromosomal mutations, thus, is an instrument of ecotypic adaptation. It is evident that these two aspects of chromosomal mutation, the production of mechanical incompatibilities and development of new supergenes, reinforce each other. There will be a steady selection for an improvement of the adaptation supergene, and this will tend to produce an increase in genic heterozygote inferiority. This, in turn, will strengthen the effectiveness of the cytomechanic isolating mechanisms. 130 EVOLUTION Evolutionary Biology. 4. Instantaneous specification through polyploidy. Polyploidy is a multiplication of the normal chromosome number. It is very widespread among plants and is an important mechanism of speciation in the plant kingdom. Stebbins, 1950, Grant, 1963. Except conifers, 1/3 of all species of plants have arisen by polyploidy. Among animals, the polyploidy is much rarer. Among animals, it occurs only in those groups which reproduce parthenogenetically. In few parthenogenetically reproducing animal groups such as lumbricid earthworms, turbellarians, and in certain groups of weevils, polyploidy is the principal method of speciation. B. Gradual speciation. Gradual speciation is the gradual divergence of populations until they have reached the levels of specific distinctness. Two modes of gradual speciation have been postulated, one involving geographical separation of the diverging populations, geographic speciation, and 2, the other involving divergence without geographic separation, sympatric speciation. 1. Geographic or allopatric speciation. Geographic or allopatric speciation states that in sexually reproducing animals a new species develops when a population that is geographically isolated from the other populations of its parental species acquires during this periods of isolation characters that promote or guarantee reproductive isolation after the external barriers break down. Meyer, 1942. The geographic speciation is the almost exclusive mode of speciation among animals, and most likely the prevailing mode even in plants, is now quite generally accepted. For example, suppose there is a large assemblage of land snails subdivided in 3 geographical aggregations or races A, Band C, each adapted to local environmental conditions, figure 10.1. Initially there exists no barrier separating the populations from each other, and where race A meets race B and race B meets race C, interbreeding occurs. Zones of interbreeding individuals are, thus, established between the races, and the width of these zones depends on the extent to which the respective populations intermingle. Now suppose, some striking physical feature barrier, such as Great River, forging its way through the territory and effectively isolating the land snails of race C from those of race B. These two populations may be spatially separated from each other for an indefinitely long period of time, affording an opportunity for race C to pursue its own evolutionary course. Two populations that are geographically separated, such as B and C in our pictorial model, are said to be allopatric. After eons of time, the river may dry up and the hollow bed may eventually become filled in with land. Now, if the members of population B and C were to extend their ranges and meet again, one of the two things might happen. 1. The snails of the two populations may freely interbreed and establish once again a zone of intermediate individuals. 2. The two populations may no longer be able to interchange genes. If two assemblages can exist side by side without interbreeding, then the two groups have reached the evolutionary status of separate species. A species is a breeding community that preserves its genetic identity by its inability to exchange genes with other such breeding communities. In our pictorial model, Fig.10.1, race C has become transformed into a new species C. 2. Sympatric speciation. The majority of authors until fairly recently considered sympatric speciation, that is, speciation without geographic isolation, to be the prevailing mode of speciation. Such a speciation is based on two postulates: the establishment of new populations of a species in different ecological niches within the normal cruising range of individuals of the parental population, and b. the reproductive isolation of the founders of the new population from individuals of the parental population. Gene flow between daughter and parental. 2. Sympatric speciation. The majority of authors until fairly recently considered sympatric speciation, that is, speciation without geographic isolation, to be the prevailing mode of speciation. Such a speciation is based on two postulates: the establishment of new populations of a species in different ecological niches within the normal cruising range of individuals of the parental population, and b. the reproductive isolation of the founders of the new population. The majority of authors until fairly recently considered sympatric speciation, that is, speciation without geographic isolation, to be the prevailing mode of speciation. Such a speciation is based on two postulates: the establishment of new populations of a species in different ecological niches within the normal cruising range of individuals of the parental population, and b. the reproductive isolation of the founders of the new population from individuals of the parental population. Gene flow between daughter and parental population is postulated to be inhibited by intrinsic rather. Gene flow between daughter and parental population is postulated to be inhibited by intrinsic rather than extrinsic factors. The concept of sympatric speciation is far older than that of geographic speciation and goes back to pre-Darwinian days. Speciation 131. Definition of sympatric speciation. The method of origin of reproductive isolating mechanisms within the dispersal area of the offspring of a single deme is called sympatric speciation. The size of the dispersal area is determined, for instance, in marine organisms by the dispersal of the larval stages. In most insects it is determined in the adult state by the more mobile sex. Since there are normally a great many ecological niches equals the constellation of environmental factors into which a species, or other taxon, fits within the dispersal area of a deme, niche specialization is impossible without continued new pollution by immigrants in every generation. Reasons for postulating sympatric speciation. Numerous biological phenomena suggest, at first sight, the occurrence of sympatric speciation. One is the abundance of finely adapted sympatric species in local fauna. For instance, there are several hundred, perhaps 500, species of bees of the genus Perdita. North America. All are oligolectic, confining their visits to the flowers of a single species or of a group of closely allied species. Many species of Perdita may be found at a single locality but never together because they occur on different plants or in some cases at different times of the year. A similar ecological specificity characterizes the species of many other genera of animals. All 6 species are perfectly adapted to the particular environment in which they occur. Could such a perfect fitting of species into their ecological niches have evolved as a purely accidental byproduct of genetics changes accumulated during geographical variation. 132 EVOLUTION Evolutionary Biology. Graphic isolation. Recent understanding of genetics of geographic speciation has shown that ecological differences can be acquired in geographic isolation and can be increased by character displacement after the secondary establishment of symmetry. Biological and host races. A necessary corollary of any theory of gradual speciation is that there should exist in nature some forms, or varieties, or populations that are incipient species. The supporters of sympatric speciation have always cited the occurrence of biological races as examples of such incipient species. Most of the early supporters of sympatric speciation adhered to a morphological species definition and assumed that the acquisition of ecological or other non-morphological species characters preceded the acquisition of morphological species characters. Any discrete population characterized by non-morphological characters was accordingly interpreted as a biological race and incipient species. Thus, kinds of animals that show no, or only slight, structural differences, although clearly separable by biological characters, are called biological races. This definition of biological race, however, includes the following heterogeneous assemblage of phenomena in it. Morphs, clones, geographic races with distinctive physiological or ecological properties, semispecies and polyploids obviously do not qualify as incipient species under the concept of sympatric speciation. Most so-called biological races are now known to be sibling species. The only biological races that might, at least in principle, qualify as incipient sympatric species are seasonal races and host races. Table 10:1. Phenomena listed as biological races. Occurrence population. Same different sympatric morphs polyploids clones ecological races host races. Seasonal races. It has been postulated by various authors that a species with a very long breeding season might be sympatrically split into two if the genetic continuity between the earliest and last breeders of year could somehow be interrupted. This occurrence of seasonal races is often cited as evidence for such a process of speciation. Host races. In many species of animals, particularly nematodes and insects, temporary strains called host races may develop on specific host plants or animals. Host races have the following characteristics. 1. Preference for a given species of host plants may and nearly always does have a double basis, conditioning, including larval conditioning, and a genetic predisposition. 2. Nearly all species with host races concentrate in a given district on one host, yet they also have the ability to establish themselves on a variety of other host plants, particularly under crowded conditions, and may have different preferred host plants in different districts. 3. Local populations teams of insects often have considerable genetic variability with respect to host specificity. Any monophagous, e.g., Microlepidoptera, solitary bees, beetles, Acmeodera, Agrolus chrysomelid calligrapha, Arthroclames, leaf diptera and other groups, or oligophagous species of insect will come in contact, during its dispersal phase, with numerous plant species other than its normal host. If the species has the appropriate genetic constitution, it will establish itself on the new. Speciation 133. Host, and this, according to the Ludwig theorem, leads to an expansion of the food niche of the species. 4TT is also found that if such a mixing of demes or local populations is prevented artificially and an inbred strain is selected experimentally on a single one of several original hosts, such a strain may become progressively less tolerant ecologically until a stage is reached when it may be difficult to re-establish it on any of the other original host species. 5. Mortality occurs whenever a strain is established on a new host. The more usual the host, the heavier the initial mortality. Among the many kinds of so-called biological races, it is the host races that have the best claim to the designation of biological race. Meyer, 1970. Means of sympatric speciation. Thus, we can conclude that the sympatric speciation is caused by the following natural situations: sibling species, monophagous species, parasites, species swarms, and the instantaneous splitting of fossil lineages. Hypothesis of sympatric speciation. Certain evolutionists have forwarded the following hypothesis for sympatric speciation, but all of these are not supported by evidences. 1. Homogamy. The concept of homogamy was postulated under the pre-Mendelian theory of blending inheritance because without its random mating would soon have led to complete elimination of all genetic variability. According to this concept, the most similar individuals of a population tend to mate with each other. Further, it postulated that monogamy would lead to homogamy. Both of these assumptions are supported by facts. Meyer, 1947, except that in a single case of birds where monogamy isolate but never leads to homogamy. In case of two kinds of geese, namely snow goose and blue goose, monogamy leads to homogamy and both are found to belong to a single species, ands are coalescents, and are found to have differences in color pattern due to a single gene, semidominant for blue Cook and Cooch, 1968. However, a mild form of homogamy occurs in many animals with highly variable adult size but that is quite insufficient for sympatric speciation. 2. Conditioning. Many workers such as Thorpe 1945 and others have suggested that the establishment of a new sympatric species population, e.g., insects, in a new niche might be achieved through conditioning. But Meyer 1947 contradicted it by showing that complete isolation of the two populations was never achieved by any of the conditioning experiment of Thorpe with insects. 3. Preadaptation and niche selection. Most hypotheses of sympatric ecological speciation postulate that dispersing individuals search actively for that particular niche to which they are best adapted on the basis of their particular genotype. There is some evidence for the validity of this assumption, since a given species usually has well-defined species-specific habitat preferences, and these are not necessarily identical for all the various genotypes of a species. Most habitat selection is, however, species-specific only in a very generalized way and is also often affected by non-genetic influences, conditioning and so forth. Only in an exceptional case will an individual search out that particular subniche for which it is specifically preadapted by its genetic constitution. The concept of sympatric speciation by preadaptation is quite typological in making the assumption that a single gene preadapts an individual for a new niche. Indeed, it would require a veritable systemic mutation to achieve the simultaneous appearance of a genetic preference for a new niche, a special adaptedness for this niche, and a preference for mates with a similar niche preference. The known facts do not support these assumptions. Sympatric speciation by disruptive selection. The method of natural selection for phenotypic extremes in a population until a discontinuity is achieved is called disruptive selection, Mather, 1955, or diversifying selection, Dobzhansky. Muller, 1940, was first to suggest that the accumulation of different sets of specific modifiers might lead to sympatric speciation. Day and Gibson, 1962, recently have studied different aspects of 134 evolution evolutionary biology. Disruptive selection, and they have found that when simultaneous selection for high and low bristle number was carried on in a population of Drosophila melanogaster, it responded with a strongly bimodal distribution within 12 generations of selection. However, when these selection experiments were repeated by other workers, similar results were not obtained. When the selection was phenotypically successful, one of the resulting morphs was of lowered viability and could be maintained only with difficulty in competition with the more successful morphs. Further, a selection pressure that would permit the survival of only two opposite extremes is unlikely under the variable conditions of natural environment. A polymorphism established by diversified selection is unlikely to lead speciation. A species would lose all the advantages of improved utilization of the environment acquired through adaptive polymorphism if it were to split into a series of narrowly specialized species. K. Jordan suggested that selection of different physiological varieties within a population can have only two outcomes, either polymorphism or extinction of inferior types. Jordan's conclusion is well illustrated by the North American butterfly Limonitis arthemis. According to Meyer, 1970, geographic allopatric speciation is the only important mean for gradual multiplication of species because sympatric speciation is not supported by strong evidences. Differences between allopatric, geographic, and sympatric speciation. The theories of sympatric and allopatric speciation agree in their emphasis on the importance of ecological factors in speciation. They differ in the sequence in which the steps of the speciation process follow each other. Allopatric speciation, sympatric speciation, reproductive isolation. 2. Sympatric speciation. Barrier removal. The theory of allopatric speciation lets an extrinsic event which separates the single gene pool into several gene pools, with the ecological factors playing their major role after the populations have become geographically separated. According to the theory of sympatric speciation, the splitting of the gene pool is itself caused by ecological factors, and any spatial isolation of the populations formed thereby is a secondary, later phenomenon. Quantum speciation. Quantum speciation can be defined as the budding off of a new and very different daughter species from a semi-isolated peripheral population of the ancestral species in a cross-fertilizing organism. Grant, 1963, 1971. The process of quantum speciation by which rapid evolutionary transition is achieved in small populations is not fully understood, but chromosomal rearrangement. Speciation 135. Translocations and change in gene regulation often play important roles. Stanley, 1979. Geographically, quantum speciation often occurs during adaptive radiation, by the release of genetic variability within ecologic islands. 4. Example, most characteristic. Example in plant is Clarkia lingulata, a narrowly endemic species of the Central Sierra Nevada, California, clearly derived from the more widespread C. biloba, and differing from it in both floral morphology and karyotype. C. Stebbins, 1976. Hampton, Clarkia biloba. Carson, 1968, reported how eco-insular radiations leading to quantum speciation has produced about 238 species of Drosophila on the Hawaiian Islands during about 5.6 million years. The quantum speciation differs from the conventional speciation in the following ways. 1. Quantum speciation is rapid, requiring only a few generations. 2. The ancestors of new species do not include a large proportion of the populations belonging to the pre-existing one and may consist of only one or a few individuals. Conventional speciation is a process of splitting, quantum speciation is a budding process. 3. Conventional speciation may be promoted by drastic reduction in population size, but this is not necessary as it is for quantum speciation. 4. Conventional selection in its entirety is either guided by or is the byproduct of natural selection. Quantum selection usually includes one or more chance events. Differences between speciation in Animates and in Plants. The pattern of speciation in animals and in plants has the following 5 significant differences. 1. A plantisemuchless complex organism than Anonymal Grant, 1971. One way of quantifying this difference is to compare the number of different kinds of differentiated cells found in the adult body. Estimates made by Stebbins, 1976, indicate that this number varies from 47 to 52 kinds of cells in flowering plants, about 66 different kinds in an earthworm, 100 to 150 in an insect, and 200 to 250 in a mammal such as Homo sapiens. Moreover, the integration and delicate balance between organs required for motility and sense perception in animals are far greater than anything existing in plants. Finally, plants have an open system of growth, based upon embryonic tissue or meristems that occur at the ends of their shoots or roots and are potentially immortal, while animal integration requires a finite fixed body size and compactness, and youth, maturity, old age and death are necessary consequences. This difference affects speciation because when developmental patterns are relatively simple, the possibility that elements from two different patterns can be combined to make a functional intermediate is much greater than when they are highly complex. 2. Because of their highly developed sense organs, animals can easily develop barriers of reproductive isolation based upon divergent behavior patterns, i.e., ethological isolation. In many groups of plants, on the other hand, particularly those having accidental cross-pollination by the wind, the possibility for building up prezygotic barriers of reproductive isolation is much less. Baker, 1959. 3. Because of their great capacity for vegetative growth, sterile hybrid plants in many groups may be virtually immortal. For instance, the Canadian pondweed, Elodea canadensis, was introduced into Clarkia lingulata. 136 evolution, Evolutionary Biology. Europe is female plant about 1840, but no males of this dioecious species arrived. Nevertheless, between 1840 and 1880 it spread by vegetative means throughout the inland waters of Europe. Gustafson, 1946-47. Similar is true for the land plant, the grass red fescue, Pistuca rubra, Harvard, 1961. Thus, sexual sterility does not cause any hindrance to occupation of habitat in plants as it in animals. In perennial plant species, particularly those equipped with rhizomes or bulbs, a sterile hybrid can occupy indefinitely whatever habitat may be open to it. Moreover, even if it has a very low level of fertility, such that less than 1% of the flowers can produce seed by sexual means, it can eventually produce many offspring that share its adaptive vegetative characteristics, e.g., two grass species, Elamis condensatus and E. triticioides, Stebbins, 1959. 4. Sterile hybrids of plants may acquire fertility by chromosome doubling or Polypellidia canadensis. Loidy more easily than animals. For this reason alone, reticulate evolution is likely to be much more common in plants than in animals. Reticulate evolution can be defined as evolutionary change resulting from genetic recombination between strains as an interbreeding population. 5. Many plant species are both hermaphroditic and capable of self-pollination, so the tuniparental reproduction by autogamy is for them a normal method of reproduction. RUAYRYO 018 DMN OK. Write an essay on the process of species. Mountain ranges are topographic barriers as they tend to limit the distribution of terrestrial animals. Barriers. NY physical, biological or climatic factor that restricts the migration or free movement of individuals or populations is called barrier. For example, the physical barriers include high mountain ranges, large water bodies between two land masses and large land masses between two water reservoirs, the difference in salinity of water, the depth of oceans, presence of very thick forests and absence of vegetation. According to R.S. Lull, 1957, and P.A. Moody, 1970, there are the following types of barriers. 1. Topographic barriers. High and extensive mountain ranges act as barriers. They tend to limit the distribution of terrestrial animals. They are more effective if they are parallel with the equator as in Asia and Europe. In consequence, there exists a marked difference between the species occupying the northern and those occupying the southern slopes. For example, the Himalayan ranges of India are parallel with the equator and are covered by snow. On the south of it, there are hot moist plains of India having tropical fauna, which resembles that of Africa. It contains elephants, tigers and rhinoceros. In the north of Himalayas, the climate and other conditions are changed and as a result the animals of that region are comparable to those of Europe. It contains oxen and gibbons. In the New World, i.e., North and South America, the mountain ranges run north to south and their influence as barriers upon the distribution of animals is very less. The mountain ranges of western United States of America exercise an inescapable influence on the control of the humidity. The moisture-laden winds from Pacific Ocean never cross 138 evolution, evolutionary biology. The mountains and are precipitated on the western side. Due to this action dry climate prevails on the eastern side. This has a great effect upon the nature of the vegetation and also upon plant-eating, or herbivorous, animals. One important topographic barrier in North America is that which limits the Mexican Plateau. The plateau itself has a temperate climate, and the fauna is similar to that of the region to the north and northwest. From the edge of the plateau the land drops away abruptly and conditions rapidly change to that of a hot, moist, tropical region, with a corresponding change in the plant and animal life, which is now that of tropical Central and South America. 2. Climatic or ecological barriers. They include the abiotic ecological factors such as temperature, moisture and light which control the survival and dispersal of animals. Temperature. The temperature acts as an important barrier for both cold-blooded as well as warm-blooded animals, because none of the two can survive the extremes of temperature changes. The effect of temperature is much more marked in limiting distribution of cold-blooded animals. For example, the amphibians and reptiles are tropical and temperate in their distribution. They rapidly diminish towards north and south poles. Among reptiles, crocodiles are tropical or subtropical. The northern limit of turtles is 50 degrees latitude. The lizards are very few beyond 40 degrees latitude. The serpents, snakes, have the widest range, but only 3 species in Europe are found beyond 55 degrees latitude and one common viper extends up to the Arctic Circle. Birds and mammals, e.g., tigers, elephants, etc., have no such limitations. Moisture. The moisture is another very powerful factor which acts as barrier, because its lack and excess both produce different types of habitats. Lack of moisture produces desert conditions. Some forms can bear the drought conditions and some not and, hence, act as an efficient barrier. The most notable desert barrier is that of the Sahara in Africa, which limits the distribution of deer. Deers are found in Eurasia but are absent from Africa except in its northern part. Straits of Gibraltar to Tripoli. The Arabian Desert also acts as a barrier for such forms. Animals such as amphibia, which require moisture for their larval life and have no great migratory powers, find even a small arid area an impassable obstacle. Increase of moisture in a region makes it. Lack of moisture produces desert conditions. Unsuitable due to swampy conditions or X's. Sieve growth of trees. For certain creatures e.g. Permian reptiles, it had become a formidable place. 3. Vegetative barriers. The forestation and the food supply, both, tend to limit the distribution of animals. The luxuriance of vegetation depends very largely upon the climatic conditions, i.e., the temperature and degree of moisture. Its prevalence is favorable to the distribution of certain types of animals and unfavorable to others. The influence of vegetation is both direct and indirect. Its direct effect is the arboreal animals, tree-dwelling, which cannot cross the regions where forests do not occur, e.g., prairies. In the same way, the larger terrestrial animals cannot penetrate through the dense forest of tropics. Thus, in Pleistocene times mastodons of the genus Dibelodon migrated from North to South America across the newly established Isthmian land bridge, but during the Pleistocene, while there was barriers 139. Several species of magnificent elephants, Elephas and Mastodon, in North America, none of them apparently succeeded in penetrating south of the southern limits of Mexican Plateau. Primates were abundant in West North America in Eocene, but by the end of Eocene they became extinct due to the change of tropical forest by deciduous forests. The lack of vegetation also affects the distribution of certain animals. For example, the primates, which are mostly dwellers of the tropical forests, depend directly upon the huge vegetation for movement and also for food, because they eat fruits, nuts, buds, etc. Further, there is a necessity of trees and shrubs for animals, which have short-crowned browsing teeth and pasturage of harsh grasses for those who have long-crowned grazing teeth. Each type of vegetation being unadapted to the other type of animals. Distribution of certain types of insects and caterpillars is also dependent upon the vegetation of an area. They feed upon certain plants and, hence, if these plants are absent from that area, those insects and caterpillars will not exist. Insectivorous birds are also dependent upon the availability of specific insects. 4. Large bodies of water as barriers. Large bodies of water such as large rivers and oceans act as barriers, especially for the terrestrial vertebrates such as amphibians, reptiles and mammals, but not for the aerial creatures such as birds and bats. Mozambique. Channels 400 kilometers wide, separating the Madagascar island from the mainland of Africa. And sea, separating New Zealand from Australia. The fauna found in Madagascar is quite different from Africa. The river Amazon, Brahmaputra, and Ganga with their tributaries limit the distribution of forest animals. Freshwater fishes, e.g., carps, garpikes, catfishes, bowfin, etc., are unable to migrate through the large bodies of salt water. 4. Amphibians. Salt water even of 1% is poisonous. They are completely absent from southern hemisphere, e.g., Australia, South America and Africa. Burrowing caecilians are confined to South America and Africa. For crocodiles and marine turtles, the seas do not act as barriers. Giant tortoises are confined to certain oceanic islands, e.g., Galapagos Islands and the islands of the western Indian Oceans, and are totally absent on the mainlands of South America, Africa and Eurasia. Large water bodies act as barriers for terrestrial vertebrates but not for aerial creatures. The serpents, snakes, except the sea snakes, cannot swim through the large bodies of seawater. Lizards too cannot cross these oceans, but their eggs may be transported to oceanic islands by the birds. Birds, except flightless birds, e.g., rhea, ostrich, cassowary, can do transoceanic migrations. Among mammals, except whales and seals, all cannot pass through the large oceans. Bats, however, are capable to cross the large oceans and are found in oceanic islands. On the other hand, large land masses act as barriers for the aquatic animals. 5. Lack of salinity of seawater as barrier. Salinity of seawater remains constant and it acts as an effective barrier for certain kinds of sea animals, such as the brachiopods, echinoids, crinoids, starfishes, squids, foraminiferans, corals and sponges. These animals tend to live in pure saline water and are totally absent from those regions where rivers enter the sea. The corals are mostly marine, and few are found in brackish and fresh water. With few 140 evolution evolutionary biology. Exceptions: all corals require water of maximum purity and salinity. They are never found near the mouths of large rivers such as the Amazon, South America, the Orinoco, Venezuela, and the Mississippi, USA, where the salinity is greatly changed. Great Barrier Reef on the east coast of Australia acts as barrier. It hinders the dispersal of animals. Further, like the salinity, the depth of ocean acts as an effective barrier. Thus, animals living on the surface and mid-water cannot move to deeper waters because their bodies would not be able to withstand the increase in atmospheric pressure and decrease in light. Similarly, animals from depth cannot survive in surface waters because their bodies will not be able to withstand the decrease in pressure and increase in light intensity. Great Barrier Reef. 6. Biological barriers. The biological barriers include preferential type of food. Certain animals depend on particular type of plants for their food, shelter and breeding, and if those plants are not present or removed, then that particular species would gradually perish. However, if such plants are available in some nearby area, they would migrate. Similarly, certain animals act as parasites and others as predators. Of these animals, availability of the host animals and the prey respectively is most essential for the survival. If the host species or prey are destroyed or removed from an area, the parasites and predators would either disperse or become extinct. RUAYRYO 018 DMN OK. 1. Describe the various barriers, which restrict the zoogeographical distribution of animals. 2. What are the barriers? How do they affect the distribution of animals in space? 3. Discuss the role of barriers in the dispersal of animals. The outcome of chemical evolution was the origin of life. Origin of life. Key kind of evolution, which includes evolution from atoms and molecules to simple and then complex substances, and from these to still more complex ones capable of self-duplication, has been termed as inorganic, molecular, or chemical evolution. The most significant outcomes of chemical evolution are the origin of biologically important macromolecules such as proteins and nucleic acids, RNA and DNA, origin of life and an environment to sustain life on the planet Earth. Historical and theorized. The question of origin of life on our planet, the Earth, has remained a most complicated and puzzling problem for thinkers, philosophers and naturalists of modern as well as ancient times. Different views have been forwarded concerning the origin of life on Earth by thinkers of different ages. Most of them are quite bizarre and merely having a historical significance. Certain most important hypotheses and theories concerning the phenomenon of origin of life are the following. 1. Special creation theory. According to the special creation theory, the life is mysterious force or vital spirit that set living things completely. 142 EVOLUTION Evolutionary biology. Apart from the non-livings, and it is originated on our planet due to some supernatural event, which cannot be studied scientifically but must be accepted on faith. Most religions of different human cultures accepted the concept of special creation in one way or other. The followers of this theory argue that all living beings on the earth were originally created by a supernatural power, God. According to Bible, for instance, world was created within 6 natural days. Plants were created on 3rd day, fish and fowl on the 5th day, and animals on the 6th day. Lastly, human beings were created, first man then woman. Adam, the first man, was molded by God from inanimate matter, clay, which he furnished with a soul, thus, breathing life into him. Objections to special creation theory. The special creation theory lacks sound logic and scientifically sound evidences, therefore, it could not convince the scientists. Charles Darwin condemned the special creation theory by saying that Earth has not always been inhabited by the plants and animals as we know now. 2. Hindu concept of origin of life. Different schools of Indian philosophy and Hindu religion have elaborated the problem of origin of life in sufficient details, but that is beyond the scope of this book. However, it will not be out of context to discuss here some ideas of Upanishad philosophy. For instance, the cosmology elaborated in the Taittiriya Upanishad describes the origin of life in the following way: from Brahman sprang ether, from ether air, from air fire, from fire water, from water earth, from earth herbs, from herbs food, from food seed, and from seed man. Likewise, according to the Mundaka Upanishad, life arose from food and from food arose the mind. Most of the early Upanishadic philosophers considered Brahman as not being something transcendental, superhuman, supernatural, or otherworldly. They did not recognize any god as the creator, controller, and designer of the world. To them, the Brahman was not a god, but the infinite totality of existence. For details see K. Damodaran, 1970, Man and Society in Indian Society, People's Publishing House, New Delhi, India. 3. Theories of spontaneous generation or abiogenesis. For centuries, the concept that life arose spontaneously from a non-immediate material was a principal doctrine of how life originated. The spontaneous generation or abiogenesis of life was visualized as beginning with either inorganic materials or with putrefying organic matter. In China, for example, even from the earliest times, there was belief in the spontaneous generation of aphids or other insects under the influence of heat and moisture. In sacred books of India, there are indication of the sudden emergence of various parasites, flies, and beetles from sweat and manure. It has been deciphered from Babylonian cuneiform texts that worms and other creatures were formed from the mud of canals. In ancient Egypt, the conviction prevailed that the layer of humus deposited by the Nile in its flood stage could give birth to living creatures. When it was warmed by the sun, frogs, toads, snakes, and mice were formed thus. Apparently, before the 1700s in Europe, it was common among laymen and scientists alike to believe that living matter could spontaneously develop from non-living materials. Infestations of worms, flies, fungi, and other organisms appeared regularly in food and organic matter without any apparent explanation. People were unaware of the life history of most of these organisms and could not know how easily eggs and spores were transferred from place to place. Disproof of the spontaneous generation theory. During the late 17th century, an Italian doctor Francesco Redi, 1626–1698, performed what are now considered classical experiments to test the validity of the theory of spontaneous generation and had the honor of being the first to come forward with experimental refutation of spontaneous generation so firmly established for many origin of life 143 centuries. In his paper, Esperienze intorno alla Experiment 1. Generazione degli Neti. 1668, he descria bseri dsel. F. Of experiments in which he l. placed meat or fish _lm, lu ll under clean muslin coverings. And demonstrated that while, and, I% flies laid eggs on muslin, beginning of experiment. Maggots or larvae appeared. Only when those eggs were transferred to the meat and equals 5 allowed to hatch. He fl. I. pound. Concluded that maggots 10L came only from pre-existing T** flea, S and were knots. Yield spontaneously generate ed by beginning of experiment any other form of material. While Redi's experiment proved that even the simple forms of life known in the 17th century did not arise by spontaneo us generation, the discovery of microbes by Van. Experiment 2. Leeuwenhoek, 1632–1723, jars covered with fine netting brought the question into the S. forefront of biological Fig 12.1. Redi's experiments. Thought once again, after all, maggots are far more complex than those little spherical, rod, or corkscrew-shaped bacterial forms observed under the microscope. So it was considered that microorganisms arose by spontaneous generation. This idea was very authoritatively maintained by the noted German philosopher Leibniz, 1646–1716. French naturalist Buffon, 1708–1788, and Englishman John Needham, 1713–1781. The decline and fall of the theory of spontaneous generation. An Italian scholar Abbe Spallanzani in 1765 tested the theory of spontaneous generation of microbes. He prepared flasks of meat broth which were boiled for several hours and then sealed. The broth remained clear for months, and when the seals were broken and the broth tested, it was shown to be free of microbes. Spallanzani's experiments were neither conclusive nor satisfying to many of his contemporary scientists. Some claimed that by boiling he had driven out a vital force necessary for spontaneous generation. When Priestley proclaimed oxygen as a necessary factor for life and experiments of Gay-Lussac demonstrated that such boiled and sealed flasks contained no oxygen, then these were seized upon as evidence that the vital principle of life required oxygen. Many of Spallanzani's successors modified the experimental procedure in attempt to quell the criticisms, but each new technique gave rise to new criticisms. The death blow to the theory of spontaneous generation of microbes was finally dealt with Louis Pasteur, 1822–1895, in the 19th century, some 200 years after the initial experiments of Spallanzani. 144 EVOLUTION Evolutionary Biology. The period of Pasteur. Pasteur devised several experimental means by which the spontaneous genera tion of microbes was disproved. The simplest and most liquid poured neck of flask. Sophisticated one was with the use of a swan neck flask. Into flask bent. He prepared a meat broth in this flask and boiled it for several hours. He then left the flask unsealed on AEB laboratory bench. Previous experiments have shown that the mere boiling of the broth did not diminish its generative capacity, so the experiment could not be criticized on these grounds. The flask was not sealed, and there was a free exchange of air with the environment, so the system did not lack oxygen. Still, the swan neck remained free of microbial contamination for months, because their swanny boiled air necks were shaped so to trap viable microbial particles forced out and to allow only air to enter the flask. After several months when he broke the neck of one of these flasks, liquid cooled I contamination by air and proliferation of microorganisms slowly air and in the fluid ensued. This simple experiment, thus, Altidus drawn line de Lucy together disproved the concept of spontaneous generation. J trapped. 4. Hypothesis of panspermia. Some scientists of 19th century assumed that 2LOMLC robes this planet was seeded from space. One of such a theory HOO equals called cosmozoic theory or hypothesis of panspermia, was developed by Richter 1865, and then supported by Thomson, Helmholtz 1884, Van Tighem 1891, and others. According to this hypothesis, meteorites travel ling through the Earth's atmosphere or strongly incandescent flask-tipped liquid. Descent only on the surface, while the interior remains contacts dust. Cold. Thus, the embryos of organisms inhabiting meteorites or the planets from which they were formed were preserved alive in the interior of meteorites, finding fertile soil on Earth, and grew and evolved to produce all microbes. The species now present. Another hypothesis of panspermia which was developed by Arrhenius, 1911, figure 12.2. Pasteur's experiment holds that spores of life together with particles of cosmic dust could be transformed from one heavenly body to another under the pressure of stellar rays. but it is doubtful that whether such spore remained viable because is especially serious danger for living embryos are short-wavelength ultraviolet radiations which penetrate interplanetary and interstellar space and are destructive to all life. From 1870 to 1940, most areas of science progressed toward a level Arrhenius, 1911, of maturity where their firmly established principles collectively created a unified picture of the world. Biology had brought life down to the chemical level through physiology and biochemistry. Chemistry had raised the elementary structure of matter to large synthetic molecules. Physics had provided the energy considerations on which reactions were based. Geology had measured. Origin of life 145. The age of Earth is a few billion years and that of the universe is close to 10 billion years. The stage was set for the idea of spontaneous generation to be given a more rigorous inspection. The first form of life was not to be a maggot, or bacterium, or even a virus but a collection of biologically important molecules, capable of producing organized forms of life through processes of natural selection. The laboratory was the entire surface of Earth, the time was in terms of several billion years, and the experimental procedure was natural selection. 5. Theory of chemical evolution and spontaneous origin of life at molecular level. Since Pasteur's time, countless generations of students have been taught not to believe in spontaneous generation. However, Pasteur's experiments revealed only that life cannot arise spontaneously under conditions that exist on Earth today. In fact, a Russian biochemist Alexander I Oparin in 1924 has proposed that conditions on the primeval Earth billions of years ago were definitely different from present conditions, and the first form of life, or self-duplicating particles, did arise spontaneously from chemical inanimate or abiotic substances. Thus, Oparin was the first to suggest that a long evolution of chemical substances occurred before life actually originated. In his book, The Origin of Life on Earth, in 1924 he provided a biochemical explanation of origin of life. According to him, origin of life occurred along the origin and evolution of Earth and its atmosphere. The primordial atmosphere of Earth had water, methane and ammonia from which as a result of a series of changes, a colloidal body called coacervate and containing a mixture of biologically important macromolecules like proteins, lipids, nucleic acids, etc., were formed. These coacervates though were not living, but they behaved in a manner similar to biological systems by being subjected to natural selection, by being chemically directive and by having the capacity of reproduction by fragmentation. Synthesis of coacervate droplets. The coacervate droplets have been made typically by combining solutions of oppositely charged colloids, e.g., gelatin plus gum arabic, or serum albumin plus gum, or clupine plus gum, or histone plus gelatin, histone plus RNA, or gelatin plus gum plus DNA. Each solution is uniform, but when the two are brought together they interact to form swarms, or clusters. When these clusters reach a certain size they separate from the solution in the form of coacervate droplets. Characters of coacervate droplets. Oparin's coacervate droplets can take up materials from the external medium and some of them show enzymatic activities. For example, Oparin and his associates have made coacervate droplets from gum arabic and histone at pH 6.2 and have included phosphorylase enzyme during the formation of droplets. When glucose 1-phosphate is dissolved in the equilibrium liquid, the droplets are found to store starch. In another experiment, coacervate droplets of some sort have been prepared with phosphorylase and B-amylase. In this case, the starch that formed from the entering glucose 1-phosphate then decomposed into maltose which diffused into the external medium where it was detected. Similarly, polynucleotides were synthesized enzymatically within coacervate droplets. These experiments with coacervate droplets, thus, show some sense of the special benefit of encapsulation of enzymes in living entities. Oparin proposed that although the coacervates which might be originated during chemical evolution, were not living, they behaved in a manner. Coacervates. Figure 12.3. Coacervate droplets. Similar to biological systems, 1. By being subjected to natural selection, in terms of a positive selection for stability as expressed through persistent size and constant chemical properties, 2. By being chemically directive, selective accumulation of material, and 3. By reproduction, by fragmentation. But since coacervate droplets lack stability, they fall apart on standing and they were synthesized 146 evolution, evolutionary biology. From polymers obtained from living systems, they no longer served as the models of protocells. Lederberg has recognized the following 3 steps in the Oparin's thesis. 1. Chemogeny, chemical evolution, 2. Biogeny, formation of self-reproducing biological units, and 3. Cognogeny, evolution of mechanisms of expressions, communication, perception and feeding. Oparin-like observations were made by an English biologist J.B.S. Haldane, 1929, about the origin of life. Both of these scientists got the ideas from the studies of biochemistry made by Sir F. G. Hopkins, and both were of the opinion that the evolution is purely chemical, the gradual transmutation of inorganic compounds into organic ones. Experimental support of O3. ITEEE, tungsten. Oparin's hypothesis B. Oparin's ideas created a trademark rash of excitement, but it was not until 1953 that some experiment stopcock for EI. Tall evidence was gathered to support withdrawing. Port Oparin's hypothesis of Spanel. SL run 5-liter flask. Edenius generation of life at most. Molecular level. I. Gases, Miller's experiment. In 1953, Stanley Miller. Then at the University of Chicago. I and a student of Nobel laureate -1. U condenser. Harold Urey, synthesized, organic compounds under conditions resembling the primitive atmosphere of the Earth. At that Time it was believed that the pre-Me 5 type atmosphere had probably been composed chiefly of water, vapor, methane, hydrogen and ammonia, latter 3 in the pipe quote dot underscore SR ratio of 1:2. Such a gas underscore 5 containing mixture was circulated through amino acids closed apparatus by steam from boiling water and subjected to an electric spark discharge 75,000, figure 12.4. The spark charge apparatus of Miller for producing organic volts between tungsten compounds from a reducing atmosphere.